Okay, hi everybody, great. So I'm really happy to be back, and I think everyone has really acknowledged the accolades to Esper again for inviting us here. It's such an honor, and it's really motivational to see such great science. I learn more here about what's going on in the US than I do back home. So it's a pleasure. Esper asked me to talk about HIV. Say that again? That is true, that is true. Don't tell that to our leader, but anyway. So it's a pleasure. Esper's asked me to talk about HIV-associated comorbidities. I'll do my best to guide you through some of the studies. I'll talk about two main studies that we've been involved in that may be of interest to the HIV epidemic, which is now in its fourth decade and will be impacting people in many ways, even though we have had great success with treatment. So if you think about the number of people who are still dying from HIV in the current fourth decade, many of them are really non-AIDS related. As you can see, about 50% in blue are AIDS related, but a number of other comorbidities are now really responsible for the number of deaths that we're seeing, and they're really related to non-AIDS malignancies, infections affecting the heart, affecting the liver, many other tissues, as you can see here. And they really are potentially driven by ongoing inflammation and some of the pathogenic mechanisms. What I'm going to talk about is something that we've been more interested in, is what's happening in the brain. As you know, the early stages of HIV really taught us it is actually a brain disease with a lot of dementia in the early stages of disease. But now, with the advent of antiretroviral therapy, we still see some impairment in the brain. It's a little bit milder, but it still persists, and it does affect daily living. It's very hard to capture unless you actually perform neuropsychological tests, and you can begin to reveal some of these cognitive deficits in the brain. So, as I mentioned, if you were to look at multiple cohorts around the world, we're looking at about 40 to 60% of individuals developing impairment, and this is attributable to HIV in the central nervous system, and a couple of events that occur that lead to damage to the brain, and that could be driven by the inflammation, or it could be driven by toxins as well. There's a lot of interest in the field of neuroAIDS today, really trying to identify useful biomarkers that could be helpful to understand the biology and the pathogenesis of these cognitive impairments. There's also really no therapy beyond the current regimens to treat people with cognitive abnormalities, and we really are trying to evaluate other therapeutic options that will help us improve the lives of HIV patients who, as you know, will continue to take antiretroviral therapy for life, and they will be developing many comorbidities that in some cases will still lead to death. So if you look at the pre-HAART era, this is before we had effective antiretroviral therapy, the incidence of these cognitive impairments was rather large. About 18% of individuals had dementia. You saw milder forms of impairment, and many people really were asymptomatic and normal. As you can see now in the post-HAART era, we still have a relatively equal percentage of people that are still normal, but you can see the numbers have actually increased for the more milder forms of disease. So you've lowered the dementia, but you've actually increased the prevalence of these minor or moderate non-cognitive disorders and some of these asymptomatic impairments, which can only be revealed by performing neuropsychological tests. And what are the features of these comorbidities? We just held a small get-together in Hawaii to get all our patients together to meet with us and talk about our research, but also to get the Department of Health to try to talk about the epidemic in the island. And one of the things that a lot of the patients always talk about are some of the problems of apathy and depression that HIV causes, and whether this is really pathological or really just the thoughts of having HIV. But if you look very carefully, there are features of cognitive memory loss, concentration, mental slowing. There are motor defects as well that also are present, even on people with a suppressive virus. So there clearly is cognitive impairment occurring in this population. And trying to understand the biology of this has really been one of our significant projects in the lab. And if you think about the cells that are vulnerable in the CNS to cognitive impairment, two cells particularly stand out to be susceptible not only to infection with HIV, but also to the pathogenesis of CNS abnormalities. And those constitute primarily the macrophages, and the microglia, which are the resident myeloid cells in the brain. They express the CD4 antigen. They also express chemokine receptors that are important for viral entry. They're susceptible to infection, and they may also be productively infected. And I won't talk into detail about some of these studies, but these are some of the areas and cell types that are vulnerable to disruption that may contribute to cognitive impairment. There are other cells involved in the neuronal system and endothelial cells that are also vulnerable to CNS defects. So if we think about the pathogenesis, there are a number of mechanisms that have been proposed by which impairment and neuronal damage occurs. The underlying hypothesis these days has really been that your monocyte population is susceptible to having viral components or HIV replication occurring. There's belief that they cross the blood-brain barrier. So, once these infected cells enter the blood, or they may be just direct viral entry as well as another potential entry into the brain, they trigger a cascade of inflammatory processes here, maybe perhaps by cytokines, which then lead to damage to your neurons here, leading to cognitive injury in the brain. So this is sort of the basic understanding that we have in the field as to how cognitive impairment evolves through an HIV-infected individual. We recently have challenged that blood-brain barrier model of entry of the virus, and this stemmed from a recent paper in Nature suggesting the existence of a brain lymphatic conduit where the virus, where at least cells of the myeloid compartment could actually enter into the brain through a meningeal lymphatic system, and that is this depiction here that we think that another route of entry into the brain could be through the lymphatic system, which is a much more porous system for cells of the immune system to enter. And in the context of HIV, I think that is quite relevant and something that we're trying to do in terms of imaging modalities to try to identify this particular conduit in the brain. Some excellent data by Tim Schacker earlier showing you by RNA scope and DNA scope the presence of HIV in the lymph nodes. Working with Mike McGrath's group here, looking at an individual who died on antiretroviral therapy, so he was virally suppressed. And if you looked at the meninges, you can actually see P24 staining. And if you use RNA scope, these are one of the first images of actual brain tissue RNA scope. You can see RNA positive cells in the cerebellum, and these cells were macrophage CD68 positive cells. So it does appear that there is potential for viral replication in the brain, even though these donors were on antiretroviral therapy. So one of the big interests right now in the field of neuro-AIDS is really trying to identify a really good marker for determining the existence or the progression of cognitive impairment or brain-associated neurological abnormalities. A lot of work has been done looking at neuroimaging, magnetic resonance spectroscopy, which looks at metabolites in the brain by imaging, has been a very useful marker to try to determine what's going on in the brain. A number of groups have been looking at microRNAs that might be indicative of impairment. There's tremendous work looking at cerebral spinal fluid and blood biomarkers. I think there'll be a couple of talks later on looking at some of the myeloid-derived biomarkers like soluble CD163 and soluble CD14 have been shown to be not only important indicators of mortality in the population on heart, but also very indicative of cognitive abnormalities. So there's been a lot of both immunological markers of myeloid activation like neopterin, but also markers of neuronal injury such as neurofilament ligand, SB100, and tau, a couple of other markers that are continuously being reevaluated in patients from multiple cohorts. There are also markers of immune activation. I think there was a lot of talks today about some of those biomarkers, IL-1 beta, IL-6, TNF, and interferon gamma. To some extent, MCP1 as well has been linked, particularly in the early stages of cognitive impairment, as a good marker of cognitive outcomes. There's been new work looking at lipids as well as potential markers of the reservoir. So we've taken a slightly different approach. This is Michael Corley in the group who's been looking at trying to understand some of the mechanisms of impairment. And one of the things that we've been interested in is looking at the epigenome. So the epigenetics is really the phenomena of chromosomal-bound heritable changes in the gene that are expressed but are not dependent actually on the changes in the DNA sequence. So we now know that these epigenetic processes can regulate essentially every gene, and they regulate that expression. And the number of studies looking at epigenetics has really been growing over the years. And the exciting thing about epigenetics is that if you think of your genome and you think about the epigenome as what's regulating the genomic expression, one of the processes is a DNA methylation, which is a modification where a methyl group is actually added to the bases, as you can see here. And that demethylation process is a component of the epigenetic changes that will occur at different sites in the genome. Another process is the histone modifications, which allows you to access, as the modification around the histone proteins is wrapped around the DNA, and that accessibility locus is another process by which HIV transcription in the latent form is actually held into latency. And there are other features of epigenetics that are of interest, but I'm going to focus primarily on DNA methylation as one of the regulatory components of the epigenetic mechanisms that will modify genome compaction and accessibility. The important thing I want to show with this slide really is that the mechanisms that regulate gene activity are really dependent on the particular cell type. It's also particularly different in different disease states, and it's also in response to physiological stimulus. As you can see here, you have the right, the arrays, the readers that sort of modify the behavior of the genome to allow you to have which genes will be turned on and which ones will be turned off. Methylation has really been very extensively studied in the cancer field. So in the cancer realm, epigenetics, particularly broadly in multiple tissues, you can actually be useful in segregating people who have actually got malignant disease from benign disease. You can see with these heat maps, there's clear segregation of these two groups. You can also look at the response to therapy. As you can see, poor responders have a very different methylome compared to those that have good responses. You can also look at the types of cancers. Again, the type of origin of those tumors can also be determined from methylomic analyses, as well as looking at the way the disease is actually progressing. So what we did here was to try to look at monocytes because they have been implicated in the pathogenic process of HIV-associated impairment, and to see whether they may play a role in the pathogenesis of HIV-associated cognitive impairment by looking at mechanisms through epigenetic studies in the monocytes that perhaps would provide a memory-like activity here in terms of either pro or anti-inflammatory process that would be driven by the pathology. So we really came up with a hypothesis that we might be able to identify a distinct epigenetic biomarker in the monocytes that would be related to impairment. And to do that, what we did was we picked up two groups of patients here with cognitive impairment, another group without impairment. This is a very small pilot study we use as a discovery cohort to see whether we would be able to tease out the differences in epigenetics. But I want to just point out what we have here was, you can see the neuropsychological score, which is a group of 8 different composite tests that were performed on these patients. This group had usually about 0.5 tends to show impairment. As you can see, these people were unimpaired by these composite tests, which is an NPZ composite score. Essentially, viral load was intermediate. Most of these patients were on heart, and they were relatively equal on many other parameters. So what we found was very exciting was just by stratifying them, looking at a methylomic profile using a 450 kb array, we were able to stratify these 11 and 10 individuals simply by looking at the monocyte differentially expressed genes. There was about 1,000 genes that were separated in terms of the two groups. We were really surprised at the tight separation. The cognitively impaired group in green had a very different profile. Each line represents a different methylation site on the genome. And you can see we identified several of them here, up to 1,000. So we were really excited by trying to uncouple each of these different differentially regulated methyl sites. This paper is now published, and you can have a look at some of the data there. But what we found really interesting was what are those differentially methylated sites? in these individuals. And what was striking, we were looking most likely to find inflammatory genes. What we found actually were brain CNS-related gene epigenetic sites within the genome, in the monocytes. So here we have BDNF, dopamine receptor, DR3, and FGF2. These genes have been shown in the literature, at least this particular site has been linked to HIV-associated impairment in the context of drug abuse. You can actually see where we found these sites within the gene body, in the promoter regions as well, and iatrogenic regions of that particular gene. So these particular sites were very differentially methylated between the two groups. And you can see very clear statistical difference in the methylation at these particular sites, which was of interest to us. So we were struck to see CNS-related genes in the monocytes that were stratifying differences between the two groups. When we looked at other genes, another one that popped up was thrombospondin 1, very tightly differentially methylated between the two groups. We found 3 sites prior to the promoter region where this particular site was differentially methylated. You can see the difference is very tightly clustered within the non-impaired group, but was a bit more broadly separated in the unimpaired group. When you now look at protein expression, transcription of this particular gene, so you've got these sites, and then if you look at the actual gene, you can see the protein expression was also very different. So though you have lower methylation, you had higher transcription of this particular protein in these cells. So this is really interesting to us. And you can see a correlation between the level of methylation and the level of expression of thrombospondin 1. So THBS1 has been shown in published papers to be a potential biomarker of plasma viremia in patients on HIV. So I think this was a very interesting finding in terms of transcription. So 2 things we found. One are genes that were linked to the central nervous system in the monocytes, and genes linked to HIV itself, which I thought was quite interesting. As I mentioned, we do look at a composite test on these patients, looking at their cognitive impairment using a number of domains that are tested to test different motor functions, attention, working memory, language, and visuospatial. These are the tests that are done. And as I mentioned, it's a composite of all these scores, give you an actual score of their level of cognitive impairment. And when we took those values and tried to correlate them with the mechanism, the methylation levels, we were really struck by the tight correlations that we were seeing with many of these sites, methylation sites, and the level of impairment. So you can see here a couple of genes, methylation sites, the level of methylation was directly correlated with the level of impairment. In some cases, they were inversely associated. In other cases, they were positively associated. So we're very struck that these methylation sites were very useful in determining the level of impairment in these individuals. We have now carried out further studies. This was the discovery cohort. We're now looking at a totally different group of patients. These patients were much older, above 50. They were in the United States. We're now looking at this in a population in Thailand where they're much younger, different diets, different environmental influences, and we're finding actually very similar data. So this is really exciting to identify each of these new novel biomarkers that may help us understand the pathogenesis of hand. So I'll summarize this first part of the talk that this is really the first evidence of a distinct immunoepigenetic signature in the monocytes that can be characterized in individuals with infection on the basis of impairment. We've noticed that these DNA methylation signatures are located at regions of the genome that are related to genes involved in the central nervous system and that are interacting with HIV. So we think these differences could be useful in assessing neuropsychological test performance, and this is sort of just a summary of the result that we can actually use these signatures to help predict or at least determine the level of impairment from the blood. So as I mentioned, this is sort of one of our strategies to try to understand the biology of HIV-associated impairment. But the other coin of the story is what can we do? Because as I said, right now there is no therapy for these patients. So Michel D'Antoni in the lab has been working on with us in our clinical program to try to look at interventions that we can use to try to ameliorate HIV-associated impairment. And two chemokine receptors that we've been interested in that are particularly important in migration, but also in terms of the functioning of the monocytes, is one, of course, is the CCR5 receptor, which has been shown to allow HIV entry into the monocytes. There is some role for CCR5 in not only in trafficking, but also in the inflammatory and replication, but also inflammatory processes that occur with a non-functional CCR5 gene in the brain. There's lots of data to show that it can actually lower many of these features. So that is one particular target that we've been looking at, and we've published some data that we actually see some signals with CCR5 blockade with the Maravirac intensification regimen. Another approach has really been to look at CCR2. This is a chemokine receptor that's also found on the surface of monocytes and dendritic cells and memory T cells. It responds to MCP1, which I mentioned earlier as another good biomarker of impairment. It actually has been shown to release MCP1, it recruits monocytes when it engages CCR2, and this whole process can actually be part of the process by which inflammatory cytokines are released and leads to the systemic inflammatory response can be initiated through the CCR2 receptor. This is sort of an understanding of the role of CCR2 in chronic inflammation, particularly as it relates to the brain. I think MCP1 and a number of studies have shown that this particular pathway could be useful in driving neuronal injury as well. So what we have hypothesized is that perhaps if we block both CCR2 and CCR5, we could find some efficacy. So we've had access to a drug called cenecrivirac. It's currently in an early phase clinical trials right now. It's an oral antagonist for both CCR2 and CCR5. As I mentioned, CCR2 is a receptor for MCP1. CCR5, as I mentioned, is useful for entering the cells. There's been published data in early as a naive individuals given CCR5 inhibitor, CCR2, in individuals. They've actually been observing a reduction in soluble CD14, but also a decrease in viremia. I must point out these people were all naive, starting antiretroviral therapy. There are some studies now in both animals and in humans looking at anti-inflammatory and anti-fibrotic properties of cenecrivirac as well. And this is sort of a chemical structure of this oral inhibitor. So as I mentioned, there are currently no approved therapies for cognitive impairment. We think that by blocking both CCR5 and CCR2, we may be able to improve impairment, potentially by decreasing monocyte activation. So we carried out a very small single-arm study. It was 24 weeks intensification study, and we had 17 evaluable patients. All of them were on antiretroviral therapy for a considerable period of time. They were aviremic, and we made sure that they weren't depressed. So that was an exclusion criteria. And we had also recruited patients who had low cognitive performance based on either a combined Z-score of 0.5 or a global deficit of less than 0.5 in one of the 7 domains that we analyzed. And we performed neuropsychological testing at entry and 24 weeks after. And we also looked at markers of monocyte activation in these individuals. This is just an overview of the patient participants. You can see, as I mentioned in our cohort in Hawaii, many of these patients are above 50 in age, mostly male. Their CD4 counts were relatively good, above 500. They'd been on heart by an average of 16 years, so they've been continuously suppressed for a considerable period of time. They've been infected for over 20 years, and only 2 patients out of this group of 17 were on NNRTIs, which has also been sort of linked, at least with efavirenz, to impairment. So I'll share with the results of the study. The exciting data that we were able to show was that after 24 weeks in this small study, we were able to show some improvement in 3 domains. Overall, the global scores actually improved significantly in this group, particularly working memory and attention. We're able to see an improvement in those particular domains in this group. We also looked at some of the immune activation markers, and this is an intensification regimen. We were very excited to see a decline in pretty much all 3 of the commonly assessed neurological biomarkers of monocyte macrophage activation. They were all significantly reduced. The highest levels, interesting, the individuals with the highest levels of these myeloid activation markers had the most significant declines, actually, in this particular intensification regimen only after 24 weeks. One of the interesting association studies that we're able to show was some of the changes that we see in neopterin, which is a very good biomarker of macrophage activation, and has been shown, at least in blood and CSF, to be linked to impairment, was inversely associated with the change and improvement in working memory over this 24-week period. It was a trend towards significance, and I think with larger numbers we may see a better trend. But we did see in the linear regression model that neopterin was very predictive of increase in working memory within this population after this 24-week period. So really just to summarize, we're very excited. We actually now have a number of biomarkers that we can utilize to try to understand the pathogenesis of hand, but also we have potential interventions that we can use. And I think these interventions may go even beyond the nervous system. I think there is a ACDG clinical trial now to look at cenocrivirac in the context of cardiovascular disease now. And I think intersecting these data as this particular drug goes forward could be useful for not only the brain and the heart, but also for other HIV-associated comorbidities. So in summary, this regimen was well tolerated in our participants. There were some mild adverse outcomes, particularly related to GI issues. But essentially, we saw improved cognitive performance. There were some concerns about practice effects, but some of these tests counteract that, particularly the learning memory. Some of those subdomain tests are amenable to practice effects. We did see marked decrease in markers of monocyte activation. And we did see that lower circulating levels of neopterin really was a predictor of the improvement in working memory. So we're excited by these data and are trying to move this forward into a much larger study. So I'm about to end here and just sort of give you a bit of an overview again of cognitive impairment. There are a number of biomarkers that are being assessed both by imaging, both by cellular mechanisms, both in the periphery and the blood. And I think the work is still cut out for us to try to understand how we end up with neuronal injury. But at least one of the early studies that we're now looking at is really targeting these infected myeloid cells, or potentially the macrophage myeloid lineage within the brain, and perhaps blocking this particular early entry stage may be amenable to a reversible state of impairment. One of the other things that we're doing now is, and I may talk about it if I'm invited next time, is work to try to move this into the non-human primate. We're very excited about studies now that are being looking at CNS pathology in the SIV model. There are a number of models out there now that are being used, and we've been working with our colleagues in Thailand to develop a model of of SHIV actually, in the non-human primate, but it's a non-accelerated model. So it's going to probably mimic more likely what we see in humans. And we're very excited with the early data, which will be published very soon. And I can talk about some studies we're hoping to integrate with work that we're doing. But essentially, as I mentioned, there are a number of models that are in development or actually active right now that are sort of focused on neuroaids within the non-human primate models. Some of them are rapid models, and some of them are non-accelerated models, looking at different strains of virus and different mechanisms by which they induce cognitive, at least CNS abnormalities. So with that, I want to really acknowledge a lot of participants in our group who've been working on this. We are also working on some studies of moving these forward to look at ways to eradicate HIV within the CNS compartment. And we have an initiative now that we're hoping to move our cure studies beyond the periphery and into the brain. So with that, I'm happy to take any questions. Thanks.